Hello, and welcome to AtheroTalk, the podcast series by the EAS Young Fellows. I'm Fotis Barkas. I'm an assistant professor of internal medicine in the Faculty of Medicine of the University of Ioannina in Greece, and I'm going to be your host for today. As part of our mission to make AtheroTalk truly global, we are privileged to speak with leading voices shaping the future of cardiovascular prevention. Today, we are honored to welcome Professor John Kastelein, one of the most influential figures in the field of lipidology and cardiovascular genetics. Professor Kastelein is Emeritus Professor of Medicine at the University of Amsterdam, where he held the Strategic Chair of Genetics of Cardiovascular Disease at the Academic Medical Center. Over his remarkable career, he has authored more than 1,500 scientific publications, including landmark papers in Nature Genetics, The Lancet, the New England Journal of Medicine, JAMA, and Circulation. His work has accumulated over 100,000 citations, reflecting his enormous impact on cardiovascular science. Professor Kastelein is currently operating partner at Forbion Capital Management, focusing on cardiometabolic innovation. He also serves as Chief Scientific Officer of New Amsterdam Pharma, leading the development of obicetrapib, a novel CETP inhibitor, and also he is an advisor to Coursera and North Sea Therapeutics. So, Professor Kastelein, it is a true privilege to welcome you today to AtheroTalk. Thank you very much for joining us.
You're welcome, Fotis. I enjoy this.
So let's begin by getting to know the person behind this extraordinary scientific career. Professor Kastelein, could you start by introducing yourself to our listeners? Who is John Kastelein beyond the titles and achievements?
Yes. So I'm a Dutchman. I was born in the Netherlands. I studied in the Netherlands, but the moment I was halfway into my internal medicine training, I was very lucky is that Michael Hayden accepted me for an MD PhD program at the University of British Columbia in Vancouver. And so I moved from Amsterdam to Canada, and I spent a couple of years there. And so I really learned about North America, I think, the best way possible. I mean, after me, so this was in 1986, 87, and 88, so a long time ago. I doubt that you were born then, Fotis. This was before your time. But in those days, I think for Europeans moving to North America, being it either Canada or the United States, was kind of a golden goal to do that. And that's where I learned how to think about genetics and how to incorporate genetics into the evidence for either protection or detriment in cardiovascular disease. And I also learned that it's not impossible if you think that you can develop something, that you might actually take something and then put a company around it and start developing that. So I learned things that were very uncommon in Europe at the time, and that has really shaped my career really deeply. I also married in Canada. I got my first child, so that child has a Canadian passport. I got another child later in Amsterdam. So my first marriage and my two children basically were born in the days when I was training for lipidology, internal medicine, and also for biotech, in a sense.
You have had a remarkable career spanning decades of discovery in lipidology. So what initially inspired you to pursue cardiovascular prevention, in particular the field of lipid disorders and cardiovascular genetics?
I have to say, Fotis, is that you always listen to people that claim that their life was one big plan, but in fact, a lot of it is luck. So I went to Vancouver. I ran because my university felt it was important to study somewhere else than just in that tiny country in the north, in the Netherlands, to go to a big country that's close to the United States. I was in the Pacific Northwest Lipid Club simply because Michael Hayden was there. And at the time, also, there was a very important group in Seattle. And Seattle and Vancouver worked closely together. And the first signs of DNA were coming. And when there's DNA, there's genetics. And so we saw in our, and I had done the Canadian medical exam, so I could do outpatient clinics at Vancouver General Hospital and at St. Paul's Hospital, where I saw patients. And then you see patients and you see family trees and you hear family histories, and then you understand that the disorder that you see in front of you, early heart attacks, very high cholesterol, must be genetic somehow. And in those days, in 1985, Joseph Goldstein and Michael Brown just got the Nobel Prize for Medicine for discovery of the LDL receptor. But that was only the first genetic disorder that was discovered in lipoprotein metabolism, and there were 10s, 20s more to discover. And so that whole beginning all came together in a time when heart disease was interesting scientifically and humanly. Lipid disorders proved to be essential for the pathogenesis of cardiovascular disease. And then genetics were essential in understanding genetics and biology, of course, in understanding how this was all shaping into something that actually led to early heart attacks and strokes.
Really appreciate it for sharing your experience. So over the years, you have trained and met many leaders in lipidology. What are many mentors or formative experiences early in your career that strongly shaped your scientific path?
I think that in terms of understanding genetics and DNA research, Michael Hayden has been completely vital to my career. But later on, of course, when I got back to the Netherlands and I started two things. I started finding FH patients, this whole organization that found 30,000 heterozygous FH patients. And at the same time, I started a clinical trial kind of endeavor, because I understood that it's very nice to do diagnosis of things and understand things, but it's even better to treat them. And the statins were just also coming up. 87 was the first statin, 88 the second, and then in the early 90s the third. And so I needed an infrastructure to do trials. And I remember my first executive committee was on the TNT trial, which tested 80 milligrams of atorvastatin against 10 milligrams of atorvastatin. It was a Pfizer trial. And on that steering committee, I met Phil Barter, John La Rosa, and all these important people. And gradually I learned, I met Stephen Nichols, Corcheret, and also Steve Nissen, of course, and then Paul Ritker. And so all that whole generation of trialists I learned to know, and I sat on the steering committee for Jupiter, for TNT, for Ideal. So I also met all the Scandinavian leaders, Terje Peterson and Mats Eriksson and Matti Tikkanen. And so all the people that were building the trial database to show that statins not only lower cholesterol, but also reduce heart attacks and strokes was built in those days. And I had the incredible fortune to be part of that. And so that's where all my trial experience started. And then, of course, after the statins came ezetimibe, and then the fibrates and the fish oils and, and everything else that came afterwards. So that's how things go in life. Yeah.
So based on what you have shared with us so far, you have witnessed and contributed to many of the most important developments in lipidology over the past few decades. So how would you describe the evolution of lipidology from the early statin era to ezetimibe and then to today's landscape of precision therapies?
It's very interesting. I think that what we have come to understand is that a drug that lowers LDL cholesterol also always lowers the incidence of heart attacks and strokes. It's like a universal principle that it is the atherogenic ApoB-containing lipoproteins that drive disease, and therefore any drug but it has to be safe otherwise, of course, can be used to lower heart disease and stroke in humans. And this is true for all the statins tested. Interestingly, there was one statin, a Bayer statin called cerevastatin, that also lowered cholesterol, but that actually led to such high concentrations in muscle that it led to real muscle problems. And so it also shows you that if you don't develop the right drug, for the right indication, that even although it's a statin and you test it in cholesterol, it can still go wrong. And so there are examples enough of that. And then ezetimibe, which also had somewhat of a rocky history, and then the PCSK9s. And then by now we kind of understand that if the target is right, the drug is safe, and you test it adequately in an adequate number of patients, there is the CETP meta-regression analysis, there is the observational data and the Mendelian randomization data, and they all point to exactly the same thing, is that if you get rid of these particles, you get rid of the risk of heart disease. So from a lot of, when I was young, there was a lot of negativity. Cholesterol is needed for life. Won't you become suicidal when you lower cholesterol? Statins have terrible side effects. They give you Alzheimer's disease. They give you neuropathy and endless lists of misery. This has all borne out to not be true at all, in fact. And also ezetimibe and the PCSK9s actually have shown to be very, very safe. And so that evolution from something that was considered dangerous and rare and only to be used in extreme circumstances, now in the latest guidelines, actually there are, we're talking about moving prevention to between 30 and 40. So much more almost primordial prevention. So our ideas about preventing heart disease and strokes have really changed enormously over the last 30, 40 years. From treating the very worst at a late stage in life to basically many people at a very early stage in life. So that is, I think, the biggest evolution. And what's also important, I think, is that all the studies support each other. It is not that the epidemiology tells a different story than the randomized clinical trials or the Mendelian randomization. There are always strange people that hook themselves on a little thing and then try to say, yeah, it's all nonsense, etc. But I think that the vast majority of solid scientists now believe that what I just laid out, that actually is the case. Yeah.
Unmet need in the management of these lipidemias. Is there any gap that we should bridge?
Yes, of course. So, what we just discussed, Fotis, is what is the ideal situation and what should be done and what scientifically would be wise to do. But in fact, there are about 30 million patients in the United States and about 40 million patients in Europe that are on a lipid-lowering drug, but are not at goal. And the goals are shifting, as you understand. We have 3 goals, 170 and 55, for different risk categories. The goals are shifting down because we understand from the data, especially with the PCSK9s, that there doesn't seem to be a threshold. It's not that suddenly when your LDL, for example, goes below 40 or something, or 30, that then everything becomes dangerous or something. So it's very interesting. It's very different for blood pressure, of course. You can't lower somebody's blood pressure to 60/20. That would be extremely dangerous. But for LDL, that is not the case. And so, yes, there still is a huge unmet need in this field. But it's being rapidly addressed with all these novel drugs. There are, of course, very good data on injectable PCSK9 monoclonal antibodies out last year. There is There are oral PCSK9s being peptides co-formulated or true orals. There are siRNAs now against almost every target. And there are a host of companies offering gene editing, which is a once and for all. So yeah, it's interesting is I thought in the last 5 or 6 years or so that we were kind of a little bit in a dead zone, that everything was kind of silencing down. But in fact, it is the total opposite. In the last 2 years, we've seen an explosion of novel data and novel strategies to do something about lipid metabolism. Yeah.
So let's change the chapters of our great discussion. And we know that one of the most fascinating aspects of your career is your role in translating discoveries into therapies. You are currently involved in the development of obicetrapib, a novel CETP inhibitor. And we know that CETP inhibition has had a complex history, if I could dare to say. So what makes this new CETP inhibitor different? And why are you or if you are optimistic about its future?
Okay. That's a very good question. First of all, I think we need to reestablish what is known about CETP inhibition. And the Mendelian randomization data are enormously strong. So there is no Mendelian randomization study that does not show that if you are born with low activity of CETP, that you have less incidence of any atherosclerotic event. So there is no discussion about that at all. I mean, all the geneticists and the lipid people in our field, everybody agrees is that the Mendelian randomization data are unequivocal. So that's interesting, is that so you have genetic proof and genetic and Mendelian randomization, and then also all the animal work. So a rabbit has CETP. It's probably the reason why the rabbit gets atheros so fast. So the rabbit is an ideal animal to test CETP inhibitors in before the days when you could knock in a human CETP gene in the mouse. And Phil Barter did everything to those New Zealand white rabbits that you could imagine. And in every instance, whether it was an RNA therapy or an oral small molecule, or I don't know, I think he even tested a monoclonal antibody, CETP inhibition prevented atherosclerosis. So the animal work is completely supportive of that. And then, of course, came the human trials. Now, the human trials are a lesson in humility, I would say, and also a very good almost Harvard case lesson, because the first drug was a Pfizer drug, torcetrapib. The phase 2 trial of that drug already showed an increase in blood pressure. But everybody chose to ignore it because they thought, well, this drug is going to raise HDL by 60%, and who cares about a little blood pressure, and let's put it in a phase 3. That was hubris. That was the worst kind of drug development that you could imagine. And so the phase 3 was a total disaster. The drug afterwards, the drug proved to end up in your adrenals, stimulated aldosterone production, cortisol production, endothelin-1 production. And so that was the worst drug on the planet for somebody who just had a heart attack. So that drug had to go out the door. And now every drug has to be tested in terms of, does it also get into your adrenal? And therefore you do all sorts of work. And I can say that our drug went through all that testing and also 24-hour blood pressure monitoring studies, and there's absolutely no signal. And by the way, the other compounds also didn't have that. So that was the first kind of thing that happened in the past and is no longer applicable to obicetrapib. The second thing was also interesting is that these drugs, Fotis, were developed to raise HDL, because we thought in those days that raising HDL cholesterol would prevent heart attacks and strokes. We now know that it's much more complicated. I, by the way, believe that raising HDL does a lot of good things. and especially in conjunction with lowering of ApoB. So that's a whole new discussion, but let's forget that for a bit. But the next CETP inhibitor was the Roche CETP inhibitor, dalcetrapib. It actually only raised HDL. It didn't do anything to LDL, nothing at all. And we now know that if you don't do anything to LDL, you're not going to get a reduction in heart attack and stroke. So that trial, DAL outcomes, effectively killed the HDL hypothesis. So just raising HDL on its own is not going to be protective in a heart disease environment. Okay, second drug out the door. Then came the third drug, evacetrapib, the Lilly drug. Unfortunately, that was not a very easy drug. So there were like 2 phase 2 trials where the drug was tested all the way up to 500 milligram per day. Remember, obicetrapib is 10 milligram tablet. So there was a big difference between the efficacy of that drug. And finally, they tested it at 140 milligram in a phase 3, but they used a direct LDL assay to assess how much the drug lowered LDL. And that direct LDL assay, we now know, especially with CETP inhibition, completely overestimates your LDL lowering. So they thought that the drug lowered LDL by like 36%, but in fact it lowered it probably by like 17%. And so after 2 years, they did an interim analysis. There was a total mortality benefit, but there was no benefit on heart disease and strokes, and they stopped the trial. Now, if they would have known that the LDL lowering was only 17%, they would have not stopped the trial, and they would have continued the trial for another 2 years. So that was the third mistake in clinical development. This is a very nice class for university when you tell this. And then came Merck and Oxford. And Merck and Oxford always has been a very, very good combination. Merck is a big company that doesn't cut corners, and Oxford, you know, the godfather of all clinical trials, Richard Peto, who taught Rory Collins everything he needed to know, was professor still in Oxford. And he said, listen, if we don't know whether raising HDL or lowering LDL is doing any good, we need to make a trial that is so big that even if it's just this or just that, it's going to give us an answer. And so the REVEAL trial with the fourth CETP inhibitor, anacetrapib, was the largest trial ever done in this field, 30,000 patients, and it had a result. In fact, it had a result. And the result was quite compatible with the CTT meta-regression, although formally it was actually better than the CTT meta-regression. Hence, I still think that Lp(a) lowering, which CETP inhibitors do, changes in particles, which they also do, and potentially raising HDL in some manner also helped the result in that trial. But whatever the explanation is for the outcome in that trial, it did work. And therefore, we thought if we can find a drug that does not have the blood pressure, that does not have the fact that it doesn't lower LDL, and then in terms of the last CETP inhibitor, the half-life was too long, it should have a normal half-life. And we found that drug. And in fact, our drug is at 10 milligram lower CETP by basically 98%. So it is incredibly more potent than anything that came before. And until now, there's no safety issue. So I think that we have the genetics, we have the biology, and we have the drug to do it. And so now we need to do the trials. And then fortunately, the co-founder of New Amsterdam is Michael Davidson. He's one of my longest and most respected friends. I think that together we did over 500 trials in this field. And so if there's anything I haven't done, he has done it, and vice versa. And so it's a wonderful partnership where we and he, of course, he's an American and he has a very high position in America. I'm a European, and the same is, I think, true for me in Europe. So it's been a wonderful journey to do this together, I have to say. Yeah.
Speaking of new drug development and new lipid-lowering drugs, from your perspective, what are the most exciting therapeutic targets or technologies of lipid-lowering therapies currently emerging in lipidology?
Well, I think that, first of all, CETP inhibition deserves a very prominent place, also because we found that there is less incident type 2 diabetes in our trials, there is less loss of renal function, and then there's the Alzheimer angle. And I think that that speaks to this comprehensive lipid modulation where there's an effect on LDL, on HDL, on pre-beta HDL, on Lp(a), and on particles. So that we need to work with that and get that drug to patients. But second, of course, there are very interesting developments on the ANGPTL3, ApoC3, and Lp(a) front. We now have finally an approved therapy for familial chylomicronemia syndrome with olezarsen, the ApoC3 modulator. There are many companies working on ANGPTL3, gene editing, siRNAs, ASOs. So that's also wonderful because for homozygous FH, evinacumab is currently kind of a lifesaver especially for kids. In the Netherlands, you can prescribe evinacumab now for very young kids with homozygous FH. And then, of course, I would really like to see how this battle between injectable PCSK9s and oral PCSK9s will pan out. The target is not that exciting, of course, because it's already 10 years old. But I mean, the application of it will spark some interesting contests in this field. So yeah, that is what I'm currently concentrating on in our field. Yeah. Yeah.
Indeed, it's truly remarkable how rapidly this field continues to evolve and always for the benefit of our patients.
Yes.
So your career uniquely bridges academic medicine, clinical research, and biotechnology innovation. How do you see the relationship between academia and the industry involving in the development of cardiovascular therapies?
Almost none of the successful drugs in our field that big pharma is selling, selling is always somewhat of a strange word, but it's prescribing, are coming from biotech. So without biotech, there's no discovery almost. So all of the novel discoveries, the long-term siRNAs, the gene editing, the base editing, the CRISPR-Cas, LNPs, all of that is biotech work. And of course, a large part of that is academia driven initially and then later taken over by biotech and fully developed. So I am an very, very strong proponent of young people like you and thinking when they discover something in genetics or in biology, how can I turn this into something that might help patients? And how do I raise the money for it? How do I put a little company around it? And universities in Europe are not easy with that. In fact, this is much easier in North America. And I think that it is a huge mistake. So I hope that the European environment will make it as easy to start novel companies in our field as it is in the United States. So that's one of my hopes for the future, because I am the living example that you can do this, still have an academic position. My academic position on familial hypercholesterolemia has not changed because I'm the CSO of New Amsterdam Pharma. And I'm still interested in ANGPTL3, 4, and 8, which has nothing to do with New Amsterdam Pharma at all. So I think you can wear multiple hats and actually co-fertilize all these developments into a solid career in this field that in the end is very beneficial to patients.
So this brings up my next question, because for young physicians interested in research, the path from bench to bedside can seem complex sometimes. So What advice would you give to young investigators who want to work at the interface between science, medicine, and therapeutic innovation?
Well, everyone needs to first finish their training in their respective countries, but then you need to look abroad, and it doesn't need to be abroad immediately to Yale or MIT. I mean, there are very good centers in Europe where it is somewhat easier than in other places so you need to find mentors that have kind of walked this path, have talked to venture capital, have tried to or have been successful in creating a company, doing something new. And so in the old days, you went to America to learn about genetics and biology. I don't think you need to do that anymore. I think what you now need to do, if you're interested in therapeutics, is to find a place in academia initially with some overlap into biotech that can help you on your way. That can really help you on your way. Yeah. That's what I would advise to a young colleague of mine.
Thank you. Thank you for your valuable advice. And before we close, we always include the reflective section we call the graveyard of ideas. Because we know that science is full of brilliant ideas that didn't work out the way we expected, but often I think have taught us something important. So could you share an idea of yours, a project or a therapeutic approach that initially failed, but ultimately helped advance the field or your research?
I think that a very good example of this is HDL cholesterol. So I was one of the people, and with me many people, that thought that if you would raise HDL cholesterol, that would lead to less heart attacks and strokes. But the dual outcomes trial showed that if the only thing that you do is raising HDL cholesterol, it's actually not going to help you. After that, HDL has been forgotten for quite a long time. And what you now see is lots of publications showing that there is a U-shaped curve for HDL, where the very high HDLs are actually dangerous. And so one of my missions in life is to show that that is total, and I'm using the word bullshit, because all of that is observational epidemiology that actually fails to control for alcohol use. And so we have an analysis that we've done together with Brian Ference in the UK Biobank that shows that there is no U-shaped curve at all if you adequately treat the data. And so this is going to be an abstract at ESC. I don't know whether it's accepted, and we are trying to write it down for the European Heart Journal. But what it shows is that if you carefully look at the distribution of HDL at the high end, in fact, there is an increase in all alcohol-related morbidity, suicides, accidents, liver disease, stomach cancer, and everything. And that by normal multiple regression analysis with controlling for factors, you cannot exclude all of that. So you need some very specific pair-matched analyses to make sure that, in fact, there is no U-shaped curve and that the protection of HDL goes all the way up till the very high levels. Now, the next question, Fotis, and then I'll shut up, is if that is true, so if truly HDL protects so potently, why is it that raising it does not seem to help? And so that question is a very important fundamental question in biology that is currently most focused in my mind. And one of the answers could be is that raising HDL only works if you also lower ApoB-containing lipoproteins, because the HDL changes the relationship between ApoB and MACE. And so that is because, and there is a theory that ApoA1 prevents ApoB from attaching to proteoglycans at the arterial wall. So that would mean that if you only raise HDL, nothing's going to happen. But if you raise HDL, if you lower LDL, that your effect might be double. And so we need trials for this, and we need more Mendelian randomization. So this is what next to my work for New Amsterdam Pharma, that's what I'm currently trying to move further. Yeah.
Professor Kastelein, thank you very much for sharing your insights and your remarkable journey with us today. Your work has shaped the field of lipidology and cardiovascular prevention, and your contributions continue to inspire scientists and clinicians, including me, around the world. So it's been a true honor to host you today on AtheroTalk.
Thank you very much. I really enjoyed this interview, Fotis, and I wish you a very nice day. Thank you.
Thank you. And before we finish, we would also like to thank all our listeners for joining us for this episode of AtheroTalk. If you enjoyed today's discussion, don't forget to follow the podcast and share it with colleagues who are passionate about cardiovascular prevention and atherosclerosis. And finally, we are very much looking forward to seeing many of you at the upcoming Congress of the European Atherosclerosis Society, which will take place in my home, in Athens, in Greece, in May 2026. Until next time, thank you for listening to AtheroTalk.
I'll see you in Athens, Fotis.